My name is Dr. Eric Smith. I'm professor of neurology at the University of Calgary, and it's my pleasure to present this poster on behalf of my co-authors shown here. Our work is titled Vascular Contributions to Neurodegeneration, Methods and Initial Findings of the Comprehensive Assessment of Neurodegeneration and Dementia Study, or COMPASS-ND. And we're going to present the methods and some of the initial prevalences of vascular lesions identified by the shared MRI core lab at the University of Calgary and Sunnybrook Hospital, co-directed by myself and Dr. Sandra Black. Our objective is to describe the methods and prevalence of vascular findings on brain MRI in COMPASS-ND based on analysis of the first 200 participants. Participants underwent an MRI research protocol as described. A core lab for visual measurements was established at the University of Calgary and Sunnybrook Hospital, co-directed by myself and Sandra Black. The MRI scans undergo a blinded review by one of two experienced radiologists, and we followed standards for reporting vascular changes on neuroimaging, or STRIVE, to make measurements of the presence, number, and location of brain infarcts, microbleeds, the extent of white matter hyperintensity according to validated ordinal rating scales, and the presence or absence of a pattern of lobar hemorrhages and siderosis according to the Boston criteria for probable cerebral amyloid angiopathy. Of the first 200 participants, we were able to analyze 197. 3 participants did not undergo MRI, 1 declined, 1 had an incompatible surgical prosthesis, and 1 did not fit in the scanner. And 2 gradient echo sequences were uninterpretable due to motion, so could not be analyzed for microbleeds. In a subset of 50 of these cases, the 2 readers underwent independent review, and we found good to excellent inter-rater reliability as measured by the kappa statistic, with values between 0.5 to 0.8 indicating moderate reliability and values above 0.8 indicating excellent reliability. This is the prevalence of lesions according to the different recruitment groups. Not shown are 5 participants who are categorized in an other category according to their clinical diagnosis. Mean age was 69 to 79. The participants with mixed and vascular MCI were older than the others. You can see there was a difference in distribution of female sex. The prevalence of infarcts ranged from only 6% in persons with subjective cognitive impairment, or SCI, to a high of 39 to 46% in participants with mixed or vascular MCI or Alzheimer's disease. The number with multiple infarcts was more common in patients with mixed or vascular MCI. The prevalence of microbleeds also differed across the groups. We found that 3% to 28% had a probable CAA, or cerebral amyloid angiopathy, pattern of microbleeds and hemorrhages. And then high white matter grade was seen in 25% to 72% of the disease groups, but in only 12% of the participants with Parkinson's disease or Lewy body disease. Overall, a potential vascular contribution, which was pre-specified by the vascular team as either the presence of multiple supratentorial infarcts or high white matter hyperintensity grade, meaning beginning confluent or confluent lesions, was seen in 13% to 83%. So in conclusion, we've demonstrated that it is feasible to make MRI measurements on the COMPASS-ND data, and that the measurements show good reliability across 2 radiologists. These measurements are available in the LORS database and can be used by other researchers accessing the data. And finally, we found that potential vascular contributions to neurodegeneration, cognitive decline were commonly seen across the groups. And in further research, we will investigate the differences adjusting for age and sex across groups and to understand whether vascular lesions are associated with severity of cognitive impairment within groups. Thank you.